Well, this is referring to the classification of ALL. It's not one disease, but many subtypes or subgroups of tumour, each of which has its own particular genetic changes, both inherited and acquired, that determine how that leukemia develops and also how it behaves and how it responds to therapy. And genetics has been used to classify ALL for many, many years now, for some decades. But until the advent of sophisticated and high-resolution genetic approaches such as genome sequencing, it's not been possible to identify all the disease-classifying genetic events. So taxonomy means a classification, and by revised taxonomy, the title was referring to the notion that in the last few years, there's been really an explosion of discoveries that's increased the number of subgroups of ALL from only half a dozen or so to over 20. So there are many threads to this, and it should be said that it's still a time of discovery and a learning process, by which I mean identifying all these new subtypes in prospective studies with contemporary therapy to determine how patients respond to therapy, well or not so well, and also the potential for new therapeutic approaches. But clearly there are major implications. So the first is that previously many of these subtypes could be detected by a simple genetic test that identified one genetic change that was common to all patients in that group. We now know that many of the new groups are more diverse in their genetics. For example, ALL is often characterised by chromosomal rearrangements that result in gene fusions that are oncogenic. And we now know that some of these subgroups have not just one fusion, but often a family of different genes that are fused to the same partner gene. So one test doesn't fit all patients, and you need a more sophisticated test such as RNA sequencing or even DNA sequencing to detect those patients. Now, what it means for the clinic, well, the first would be risk stratification. So we've used genetic changes that define these subgroups for many years to initially assign risk, good risk or poor risk. So for example, rearrangement of MLL or the Philadelphia chromosome or hypodiploidy have been associated with high risk features and poor outcome. We know that some of these subtypes, at least in the data that we have in hand at the moment, are also associated with risk. So at each end of the spectrum, for example, rearrangements of a gene called DUX4, a transcription factor, we now know is associated with very good risk, both in children and adults. At the other end of the spectrum, there's a group of patients that have kinase-driving lesions. It's called Ph-like leukemia, that's associated with poor risk. And there are some other subgroups as well that are also associated with poor outcome. The second major point here is that there are clearly opportunities for targeted or tailored therapeutic approaches, whether it's tailoring a particular drug to a mutation or a drug to a pathway that we know is deregulated in some of these new subtypes of leukemia. That's very early for many of these subtypes of leukemia, and it's really at an experimental stage where many groups are modelling the leukemia, either making experimental models from the ground up in cells or in animal models, or using avatars, using xenografts of human leukemia to test the potential efficacy of new treatment approaches. Will they be effective and implemented in clinical care? Well, some of the drugs that are potentially useful are already FDA approved. There are new opportunities for drugs such as histone deacetylase inhibitors, for FLT3 inhibitors. So clearly there's the potential for using these agents if one can deliver the molecular diagnostics. For some, time will tell, I think. For some of them, we don't quite know what the best therapeutic approach will be, but knowledge is power, I would say, and would suggest that until we fully understand in large cohorts of patients the significance and potential for targeting, we won't then be able to understand which patients could benefit from newer approaches. I think you could envisage that or many different approaches to tailoring therapy, whether it's an adaptive trial design either at diagnosis or at perhaps early poor response where one then intervenes preemptively with a new agent. And, clearly this is a good point because as we carve the pie into more and more smaller segments, the absolute number of patients that fulfill the criteria for one of these subtypes becomes progressively smaller. We're no longer talking about 20-25% of patients that have a subtype, but it's more single digits. And so one does have to think about whether you have a single trial that encompasses most or all patients, that has that adaptive design with perhaps multiple agents given at a particular time of therapy, whether it's poor response or upfront, or whether you think about cooperative studies or collaborative studies where everyone is genotyping with some approach to identify patients, and then in a larger trial structure they also go on to a single agent. And I think both of these approaches are being used. There are examples from the US. For example, at my institution we have Total Therapy 17, which is really a frontline study that encompasses virtually all patients at initial diagnosis, where some but not all of these subgroups are now being used to tailor therapy. All of them are identified by sequencing, it should be said. And there are other studies that are on the horizon, such as Altogether, and some other studies where there are larger cooperative groups with larger accruals that will, it's expected will progressively start to implement some of these newer subtypes into decision making and targeted therapy. So this is clearly the most important question that you could ask. It's also the most difficult to answer because, again, this is an early stage for many of these discoveries. And secondly, few places have the level of resolution or sophistication in genomic testing to detect all of these mutations, rearrangements, and other alterations that define these groups. So I think the message at the moment for the practicing clinician would be to be aware, to understand the changing landscape of genetic changes in leukemia, and to discuss them with experts, with their diagnosticians and pathologists, to ask what the options are for diagnostics for a newly diagnosed patient, or indeed a patient that's responded poorly, or a patient that presents with relapse. Because, again, knowledge is power, and I think if people are aware of what these changes are, they'll start to be able to seek out places that can provide that level of diagnostics. Here at the meeting, it's been a major topic of discussion. There's been a lot of people asking, well, how do I get this information, and how do I get patients sequenced? And depending on what information is sought, what subtypes people want to identify, and what agents people want to use, then different approaches can be used. There are actually some fairly simple approaches using conventional techniques. Good examples include flow cytometry for rearrangement of a gene called CRLF2, which is one of the most important subgroups of PH-like leukemia. There are FISH approaches, fluorescence in situ hybridization, which is again a standard test that can detect many of the rearrangements, and there are already commercial assays for many of the new partners of rearrangement, be they in PH-like leukemia or some of the newer subtypes. Many of the additional new subtypes are also rearrangements that often involve a single gene, and so they're highly amenable to a FISH-based approach as well. So it doesn't always require the most intensive or sophisticated sequencing approach. Some of the more standard approaches can be used to detect these lesions as well. And it's likely that it's already happening that many groups and laboratories are bringing these tests online. And I think over time we'll see a rapid shift to the more widespread use of sequencing at diagnosis as the costs have come down and the ability to rapidly analyse the data becomes more commonplace. The only other point I'd make is that these discoveries, at least from my perspective and my research, have been based on many patients from many cooperative studies, St Jude, the Children's Oncology Group, and a number of adult cooperative groups from the US and also Europe, and so I'd acknowledge them for facilitating this work.